Morning or good afternoon, everybody, whether you are in Quebec or Italy, and welcome to the Biobanking Roundtable discussion organized in collaboration with CORIS and University of Padua in Italy, and Gmp Québec and Fonds de Recherche Québec en Santé on the Quebec part. This is the fourth roundtable this week as part of the CORIS Winter School series, and it is broadcasted on the YouTube channel on the CORIS site. My name is Stéphanie Lafontaine. I'm the Vice President of Scientific Affairs at Gmp Québec. A few words on Gmp Québec. We are a not-for-profit organization with a mission to fund, catalyze, and foster the development and excellence of genomic research to accelerate the discovery of new applications for genomics and health. And as part of our platform services, we are also operating a biobank infrastructure that currently store biological sample from the Quebec population-based cohort Cartagen, and is also the provincial repository right now for all viral samples collected by public health authorities at screening centers during the COVID pandemic. So before we start, I would also like to thank our co-organizer on the Quebec side, Fonds de Recherche du Québec en Santé, which is the Quebec funding agency for health research. Its mission is to promote and provide financial support for all areas of research in the field of health, to support the training of graduate students and careers of researcher, and to fund the research centers and thematic networks. I will now invite Professor Girosa, CORIS Scientific Director, to say a few opening remarks on behalf of CORIS.
Thank you, Stephanie, and it's my pleasure to welcome all of you to this round of this winter school. Today, the topic is devoted to the biobanking, and I'm sure and I hope that all the speakers and presenters of the previous days and the previous tables really enjoyed to share their expertise, their experience, their project among them. So, as I already mentioned on the opening day of this winter school, it's a special year for the University of Padua because we're celebrating the 800 years anniversary of our university, even if we look a little bit younger, the 8th centuries. Having said that, I move again to Stephanie for starting, so for the sake of time, to start the biobanking table for today round. Stephanie.
Thank you, Professor Girosa. So I will be moderating the first part of the roundtable for the presentation part, and the discussion portion today will be moderated by Dr. Anne-Marie Mesmesson. Dr. Mesmesson is the Associate Scientific Director for Basic and Translational Research at the CHUM Hospital Research Center in Montreal. She has been the Scientific Director of the Provincial Cancer Research Network, and also a founding member of the Canadian Tissue Repository Network. Also today, we are very fortunate to have 7 expert panelists from Quebec and Italy to cover various aspects of biobanking and also the central role, of course, it plays in health research. We are planning to discuss various topics of common interest, such as infrastructure, data challenges and databases, standardization, and also, of course, future trends and application in biobanking. So we do hope that the exchanges today will foster sharing of best practices and also spark new ideas for collaboration. And for the first part of the panel, each panelist will have 10 minutes to introduce themselves and give a brief overview of their research interest in relation to biobanking. I think all the presenters tested the tool on the Google Meet, so you can share your presentation yourself, or you can ask Georgia to present the slides for you. So let's start with our first presentation today from Anne-Marie Mesmesson, Professor of Department of Medicine at University of Montreal. So up to you, Anne-Marie.
Thank you very much. I would ask you to share my presentation. So it's my great pleasure today to be able to talk to you about 2 initiatives. One is a national initiative on excellence in biobanking, and the other one represents the Cancer Research Network of the FRQS. Have the next slide. So the type of biobanking that we're concerned with is clinical biobanking, which is a little bit different from population-based biobanking. And within the clinical biobanking, it is sandwiched between, on the one end, medical treatment, and where we then consent participants to be able to provide samples, and medical research, where we then provide those samples to the research community. And within the life cycle of biobanking, which can be monitored in minutes or hours or days, we have the first processing of samples which come to the laboratory once they're required in the hospital. We, of course, have annotation and storage, but we also have quality control and processing that needs to be done on those samples. And over time, then, we have to release those samples to the scientific community. Could I have the next slide, please? So biobanking isn't just simple data systems, annotation, storage, retrieval, and biospecimens, which I've talked to you about, but it also concerns governance issues, ethics and privacy of those who participate and provide samples to the biobank. It is important to have elements of education and safety within our procedures. And of course, we have to work on how we enroll and consent participants. Can we have the next slide, please? So really, the importance of biobanking is quality specimens to support the translational research that's to be done on this. And to this end, the Canadian government has supported what we call the Canadian Tissue Repository Network, formed in 2004, which was initially focused on oncology, but whose practices have now expanded to other fields. It was initiated by leading biobanks, mostly provincial, and the RR Cancer was one of the founding organizations for this network. Next, please. Now, within the activities and products, there are some that are very straightforward. Of course, we're trying to address the issues of variations in practices, and we want to minimize the impact of different practices on the quality of the biospecimens and biorepositories for research. So, and part of the solution was to create policies and standing operating procedures that would be available to Canadian researchers. Next. So these cover a wide variety of activities, ethics-informed consent, but also education material release, and then all of the procedures that one might have to entertain if one is has a professional biobank, which includes administrative issues, recruitment, records, safety, quality assurance, and of course, material handling and release. Next. In terms of the quality control, it's not just the specimen quality that's important, but it's also the quality of the data. And we have to be able to follow all of the activities within a biobank. For this reason, after an environmental scan, it was decided that we needed to develop our own software that would allow us to track all of the important events in biobanking. And for this, we created the Advanced Tissue Management System, or ATIM as we call it, which is used widely of biobanks across Canada, but is based on open software and also has international installs and is available to all. Can I have the next slide, please? So some of the features of this is its ability to be highly customizable. It can support international languages. And it has a modular design, so it's easy to incorporate new sample types. For example, you'll hear from Dr. Woodsie. Many biobanks now also collect the microbiome. It has the ability for developer tools, so you can actually customize it to meet local requirements. It has a full audit trail, so you can see who has captured the data and who has modified it. And you have administration abilities to restrict the data access to different fields. So who can edit, who can read, and who can delete, for example. Next. In terms of its infrastructure, it has different modules that allow it to perform the tasks. So of course, it has the ability to capture clinical annotation and data associated with consent. It has an inventory management. Importantly, it has a query tool, which is important because often in biobanks, what we're trying to do is we're trying to match a study design with a particular set of clinical features and types of biological samples. So you need to be able to query the entire biobank to identify appropriate samples. It has the administrative functions, of course, where you can define your groups and users. It can be customized so that you can create specific drug protocols. Can I have the next slide, please? It also has the ability to customize your storage layout and do the type of drag and drop so that you can define for the entire box or a layout things like temperature and labels. Importantly, biobanks, one of the important roles, most important role of biobanks is, of course, to give samples to studies and projects. So it's important to be able to track all of the paperwork associated with these studies, whether it's the ethics, the material transfer agreements, the shipping. And so there's an ability to create these studies. Of course, it can be linked to whatever standard operating procedures, and it has an order management in order to release samples. Could I have the next slide? In terms of ensuring quality, in terms of best practices, the CEDARNet has also created a biobank certification program, which is in 2 phases, a registration and then a certification completion. It's largely based on education and best practices with the underlying principle that a well-educated staff it will conduct quality biobank, and there is also a paper audit of all of the necessary permissions and standard operating procedures within the biobank. Could I have the next one, please? So in terms of education, it's a comprehensive package with 9 modules. The first one, which is the basics of biobanks, which all members who are associated with the biobank have to complete. And then there is a series of different modules that are completed by different members of the biobank, depending on their role within the biobank, that covers a wide variety of topics that are relevant to biobanking, such as governance, ethics, quality management, collection, storage, and distribution. This program is being endorsed by ISBER, the International Society for Biological and Environmental Repositories, and this program has been used widely internationally. Australia has duplicated this certification process, and many biobanks, whether in Canada or exterior, have attained CEDARNet certification. Next, please. At the provincial level, we have a network that is focused on oncology, which is divided in 4 research axes. One that is focused on translational studies for solid tumors, one that is focused on leukemia, and one that is focused on experimental therapeutics. But of course, there is a lot of crosstalk between these 3 networks. It covers a large number of scientists and clinician scientists that are part of the hospital-based research centers, and as well as students. Next, please. One of the activities that's important in biobanking from the network is the ability to have a harmonized consent throughout the province. So patients from the different teaching hospitals sign essentially the same consent form. We have recently updated it after 18 months of consultation with the ethics board across these centers. It is, of course, based on Quebec law. Importantly, in its most recent modernization, it includes elements of the genomics and data sharing, which are more common now for biobank samples. It includes the biobanking of live samples, so not only of cell lines, but patient-derived xenografts, of course, available in English and French. And because of the pandemic, for the first time, we've been allowed to do remote consenting. So normally in clinical biobanking, the consenting process is done within the clinic. And now we are able to contact them by phone. And so it required the development of elements of remote consenting, which includes the scripts. Could I have the next slide, please? So just to talk about two of our axes, so the for solid tumor banking, we have, although initially we focused a lot on biobanking samples at time of diagnosis, as we evolve, we are doing more fit-for-purpose biobanking, which includes longitudinal studies or specific studies associated with clinical trials. As I mentioned, we have a large portion that's developed for live biobanking. So whether it would be cell lines, tissue slices, microdissected tumors, patient-derived xenografts or organoids. These models have all been very important for the drug screening programs we have in the province. And these is a central resource that supports multiple projects. And while historically we focused initially on breast and ovarian cancer because those were at the time when the network was started, the most active biobanks, now we have greater than 30 different affiliated biobanks within the province, all focused on different tumor types. Next, please. In the leukemia biobank, this is a bit of a different model. It's a centralized biobank where samples are sent to one of the hospitals, and they're focused, of course, on identifying biomarkers of diagnosis, but also looking at the identification and for targets that would permit new therapies. Could I have the next slide, please? So just to give you an idea of the volume of activity of the network over the last 4 years, so we have thousands of breast and ovarian cancer patients that have been recruited, nearly 1,000 leukemia patients. In terms of the projects that have been supported, there are over 306 funded projects that have received samples from the biobank. In terms of new grants, during this period, there are 176 that were obtained, over almost 1,000 publications that have been related to samples produced by the biobank, and just over 500 conference presentations. And can I have the last slide then, or? So I'd like to thank you for your attention. Happy to answer any questions, and this provides you links to the different sites that can be accessed. Of course, the CDNet site, the biobanking.org, which is where the certification is, the ATOM software, which describes the ATOM software itself, and it also has education and YouTube for learning to use the software, of our Provincial Cancer Network site, and the Leukemia Bank site. Thank you.
Thank you, Anne-Marie. That was a very comprehensive overview of state-of-the-art tools for biobanking. We will keep the question and answer period for the discussion part of the meeting, just to let all presenters share their 10 minutes overview. So please keep your question for Dr. Miss Masson for the second part. I will now invite Dr. Martina Pigatzi, Professor of Maternity and Childcare Clinical Nursing at the University of Padua School of Medicine, to present her 10-minute overview.
Hello, everybody. I would like to share my screen. No, I'm not able to do so. Georgia, can you please share my slides, or let me the option to share mine, because
I think I can do it for you if you want.
Thank you.
Because I think I have it. I have the PDF.
Yes.
Just a minute. Let's say open with Adobe.
Quindi non avevo il permesso, forse.
No, veramente.
Okay. Yes. So, good afternoon. I'm Martina Pigatzi, and I'm an associate professor at University of Padua, working at the Department of Women's and Children Health, that is part of the university, but also of our hospital here in the region. I work at the laboratory of the pediatric hematology oncology, which is part of our unit where we do cancer for children with cancer. And next. Okay, so our unit is made of a clinical unit. Then we have the diagnostic lab. We had also labs for research, and we are processing hematopoietic stem cells for transplantation. We are the reference lab for all the Italy, so for the nation, since we are working together with an association, which is the Italian Association of Hemato-Oncology Pediatrics. So the association is called IEOP, and we are the reference lab. This means that every day we collect samples from more or less 58 centers, hospitals in Italy, including our islands, that send samples to our center for a diagnosis. And we are the reference lab, and I'm leading the genetic unit, and we collect samples, perform diagnosis, and also for all the material which is not used for diagnosis, we are going to biobank it. And so for this reason, we have the largest and the unique Italian biobank for pediatric cancer. Next. So our focus, the unit, but also the lab, is to improve cancer patient survival. And what we do with our samples is make a diagnosis, performing a very wide characterization of samples we received, which go through immunophenotype, molecular biology, cytogenetics, and then with the left material, we word them and participated to several projects for performing disease modeling and for doing retrospective studies that are useful for the identification of molecular biomarkers or biomarkers for new targets or for drugs, for new drugs identification. Next one. So in our lab, we are performing all these activities. So we have also the storage of slides. So because we are the morphology reference group, we are doing flow cytometry. And so we in all the data regarding flow cytometric analysis. We have a lab for circulating tumor markers. We do phosphogenomics. We have molecular genetics. We have cytogenetics. We have a group for experimental pharmacology. We have an affirmatic station for expression profile. We have mice and zebrafish facilities, and we perform also next generation sequences. And also we have a lab for therapy for inherited disease. And we have the biobank for all this activity and for all the samples we collected from whole Italy. Next. So our diagnostic laboratory is under a complete quality management system, and this is based on ISO 9001, and so the lab is certified with this, but also we are accrediting in the ISO 15189. So our quality management system covers all requirements appropriate for GCP and GMP. The system is administered by a quality office and a quality manager, and all the other senior managers are accounting for appropriateness and adequacy of data storage and also data use from the others. So our clinical and biological data are both written and we have electronic records which are managed and archived. Records are kept indefinitely unless otherwise stated by contract. The record and locations have restricted access, and the records related to the lab and department work and analysis are stored in PDF and secure locals. We have a quality management from a quality manager administrator, but we have also an IT administrator that is ensuring for this type of control. Electronic records are part of a backup files that are stored off-site location. Next. So this is our biobank. It was born in 1994, but during the years we were collecting much more and more samples. In this we have 10 tanks for cryopreserved viable cells in the MSO, so more or less 182,000 cells. We have a bank of DNA and RNA. We have more or less 200,000 tubes, and also have a smaller biobank of protein lysats and also of sample serum. Next. How we used, and we are the reference lab in Italy, we work as a IELP bank, and so inside IELP we have several groups with coordinators. We discussed projects and then perform several collaborations all around the world. By changing and exchanging samples, these are used for international collaborations, but also among our networks for different diseases. So we have lymphoblastic leukemia networks, acute myeloid leukemia networks, and so on. And together with all the groups that are around Europe, but also in US, we send and share samples for the harmonization of our procedures, and we are trying to have common SOPs. And most of our collaborations and the samples that are stored in the biobank are used for performing large retrospective studies or to create the validation cohort that would support data created by the other labs. So this is the last very big collaboration, which is we are part of a European consortium, which is called EUPAL for leukemias, and this consortium is working in together with the COG, which is the biggest hospital for pediatric cancer in US, and with also the Leukemia Lymphoma Societies, who is sponsoring this great job that is performed along several groups. But we have a lot of other collaborations with other European centers, national and international groups. Next. Yep, this is the last one, only to underline that the goal of our group, but also of IELP, so the work involved in pediatric cancer, is to give the opportunity to increase our knowledge about diseases. And so whole samples have been also involved in several basic research studies where we created a three-dimensional model, where we are creating also disease and models or PDX, so patient-derived xenografts, and also are used to improve our diagnostic tools. So we are moving to advanced diagnostics with omics integration, and everything is made in order to find new targets that could be for diagnostic, but also for new drugs and for clinical improvements and treatment improvements for our children with cancer. Thank you.
Thank you, Dr. Pigatzi, for this very nice overview of biobanking in action and large-scale collaboration. I'm sure there's a lot of parallel with activities that are ongoing in Quebec and Canada in general. I will now invite the next presenter to give our presentation. The next one is Dr. Martina Pigatzi. She's the Director of Innovation at EMA Quebec.
Yes, thank you. So is it working? Do you see my presentation? Okay.
Yeah.
Yeah, okay, that's perfect. Okay, so thank you. It's a pleasure to be here to participate in this roundtable. So my name is René Bazin. I am Scientific Director at Medical Affairs and Innovation at EMA Quebec. And well, you will realize that EMA Quebec is not a research institute, so our main mandate is not to prepare biobanks, but to bank products for the needs of the Quebec population, for the patients. And so I will give you an overview of all the products that we bank. So we, of course, are responsible for managing blood products, but also other biological products of human origin. And we really want to become a strategic partner for the Quebec health system. So in particular to help also the research efforts that are done in the province of Quebec. So our sectors of activity cover the blood products, the stable products, which are proteins derived from human plasma, such as coagulation factors, albumin, and immunoglobulins, but we also have activities around stem cells, human tissues, and mother milks. And so just to give you a timeline of IMA Quebec, so our organization was created in 1998 and in 2001, and so we started with the blood products, but soon after, in 2001, we created the tissue bank, human tissue bank. 2003, we created the public cord blood bank, and finally in 2014, we created the public mother's milk bank. So for all of our activities, we follow the same pattern. So we recruit donors, we collect the donation, transport to our facility, we perform analysis to be sure that everything meets the regulatory requirements for the products. We prepare the products, so for blood, there's separation, but tissue, we recover the tissue. We prepare also the cord blood and the mother's milk. We store the products, and then finally we deliver the products depending on the patient's needs. So everything is highly regulated. So we have SOPs for every step. We have a quality assurance program. We have all kinds of data management. We have internal audits, external audits by Health Canada. So everything has to be really highly regulated and must comply to all regulations. Okay, so just to give you an overview also of the size of our organization. So for the blood products, we deliver over 300,000 blood products every year to the hospitals. For the stem cells, we have our public cord blood bank in which we have over 12,000 units that are banked. And in 2020-21, you can see that we distributed 22 units. So this bank is for patients, for transplantation, but as you can see, there are much more units that are banked compared to those that are distributed. So there's a potential to use some of these units for clinical research, and in fact we are participating in some clinical projects, for example, for stem cell expansion before transplantation. So there's a potential to have some access to some material for doing research, and all our activities for the public cord blood bank are FACT and WMDA accredited. We also have a service of cryopreservation of autologous stem cells. So the stem cells are collected from the patient at the hospital, and the product is sent to IMA Quebec, where we process the sample and we cryopreserve. And so as you can see in the right column here, in 2020, we have cryopreserved 979 bags of cells, and we return to the hospital when the patient is ready to receive his own cell back. So we return around 600 bags. So there's a potential also. There are some unused material that cannot be used for research because we don't have the proper consent, but this is something, maybe there's a potential to have access to more material if we put in place proper consent. Then we have our human tissue bank, and as you can see, we collect over 700 donors every year, and we bank ocular tissues like corneas, eyeballs, heart valves, arterial tissues, musculoskeletal tissues, and also skin. And once again, and I forgot to mention, for the cord blood and also for the human tissue and human milk bank, we included in the consent for donation and also a consent for research use if the product cannot be banked. So if the product cannot be used for banking for the patients, it can be used also for research purposes, but we then need to have a evaluation for the ethics committee, from the ethics committee, but at least we already have the consent from our donors. It's the same for the mother milk bank. So we have over 900 donors that take their milk, and they bring that to the different centers at IMA Quebec, where then we process the milk, we store it, and then we can distribute it for the premature baby needs. Finally, we have two research biobanks at IMA Quebec. So the first one is called BARCO. It's a small bank. It was created in 2015 to provide mesenchymal stromal cells that were derived from umbilical cords for clinical research. And the intention was to do a clinical study for the treatment of GVHD in pediatric patients. But unfortunately, we were not able to do this clinical trials. But still, we have some cells remaining. So we have 85 cryovials that are available for the researchers who would need these cells. And finally, we started in 2021 a bank that we call PLASCO. It's a plasma biobank, and for that we really took advantage of our operation. So our regular operation, we are collecting a lot of plasma every year. So everything is put in place to accept the donors and to just perform the donation according to the regulatory requirements. But then we have added a consent for participating in this biobank. So we collect a 3 ml of plasma at the end of the plasma donation. And so we bank these samples for research on COVID infection and vaccination. And so as of February 2022, we have more than 94,000 samples from 15,000 different donors. And you have to understand that these donors come, some of them come every week or every 2 weeks. So we have a very good collection of longitudinal samples that can be made available also to researchers to do research on COVID. And so that's about it for IMA Quebec. So thank you.
Thank you, Dr. Bazin. That was quite interesting and a different perspective on biobanking with a different reality also, but also forward-looking in terms of research. And I really like the Plaskov Biobank idea. I think it's a great opportunity for research on COVID. So thank you for the presentation. I will now invite the next speaker, Dr. Stefano Ferrari, which is the head of research and development at the Veneto Eye Bank Foundation, to give the 10-minute overview.
Yeah. Can you see my presentation?
Yes.
Okay. Well, thank you. Good afternoon, everybody, and good morning to the speakers in Canada. Well, I'm actually replacing my director, Dr. Ponsin, who was supposed to give the talk on my behalf, but is busy in a parallel meeting of the Italian corneal transplantation exactly as we speak right now. So I will do my best to present who we are and what we do at the Veneto Eye Bank Foundation. Well, this is a beautiful picture of our eye bank. We are based in mainland Venice, and these are some of the, if you want to get in touch with us, these are our website details, so fbov.org or research.fbov.org, or you can follow us on our LinkedIn profile. Well, we are the largest eye bank in Europe. We collect more than 5,500 corneal tissues every year, and we distribute approximately, say, 4,000 tissues, 4,000 corneal tissues for transplantation. We distribute them to, well, in Italy, of course, and also, but also to many European countries like the UK, Portugal, Germany, and many others. Just to give you an idea of how much we distribute, take into account that basically we in 2019, we accounted for the 54% of all the corneal transplantation done in Italy. And in 2020, during the pandemic year, we distributed to, basically our corneas accounted for 63% of all the corneal transplantation done in Italy. This is how much this graph shows to you how much we've grown from back 2007 to 2019, with the difference then in 2007. Basically, we only distributed whole corneas for transplantation, and now what we distribute is mainly pre-cut and pre-loaded corneas for transplantation. And this is very much, everything is due to all the research that we carry on, thanks also to the tissues that are not suitable for transplantation and that we can use for new products, for validating new products and devising new products. So on top of being a bank for providing tissues to the surgeons for transplantation, I would call ourselves as a sort of disease-oriented biobank. Disease because basically we are working with collaborators who are mainly involved in cornea and retina, and in general in the eye disorders. And the sources of our biological samples for research and for training are basically we have basically two sources. One is healthy subjects, so the corneal tissues and the eye bulbs that we take from the donors that basically come to our eye bank. And the other one is diseased tissues that we get from eye surgeries. So we collaborate with many surgeons. And they give to us tissues from patients that they operate, and they give to us leftover from the surgeons. But then I will come back to that later on. To talk about the first point, so the healthy subjects, the donors, basically out of the 5,500 corneal tissues and eye globes that we normally collect, there are approximately 1,000 tissues that are not suitable for transplantation and that we currently use, 1,000 per year, that are not suitable for transplantation and that we use for validation studies and training of eye bank technicians and surgeons. And what we use them for? Well, the majority of this tissue that get used for collaborating with scientists, with surgeons, with clinicians, in order to basically try new techniques, in order to validate new tools or machines, of course in the ophthalmology field, to run some hypotheses for strategic therapeutic approaches towards diseases affecting the eye. And one of the main basically, I think that we are very focused on doing research work, collaborating with people in the networks that we have, and try to publish paper in peer-reviewed papers. This is really a focus of our center. On top of this one, we also provide corneas for training eye bank technicians and surgeons. We have a wet lab facility in our building, and you can see some pictures on the top part of the slide. And you can see that very often we invite a technician from other eye banks or also our own eye bank technicians, and we test new devices, new tools, or new techniques for making corneal transplantation better or easier for the surgeon. We also provide corneas to scientific societies to run their own wet labs during conferences of meetings. And these are some examples of the last wet labs we've held with corneas from donors. We also collaborate with companies and with pharmaceutical companies for the development of like a sort of alternative methods to the use of animals for testing, for example, new drugs, new eye drop formulations, to see whether there is some ocular toxicology, and we have some machines that sort of mimic the physiology of the eye. So the cornea is basically put in this machine, and it is in a sort of natural environment where we can test the drugs and try to see whether they are any good or any better when compared to gold standards. So this is another important part of our job. And then, I mean, starting from a few years ago, we also, I mean, basically, we normally always think about the cornea because this is what we were raised for. I mean, thinking about the cornea as an eye bank. But we've also thought that it would have been a good idea also to look at the posterior part of the eye. So we have started biobanking human retinas and human retina and choroid and optic nerves for what we think might be the next big leap for the eye banks, which is the transplantation of retinal tissue or retinal pigment epithelial, retinal pigment epithelium. So this is a nice idea that we are trying to have. So since on top of collecting corneas, we also collect eye globes, we thought that, I mean, it would, instead of throwing away all the rest of the eye globe, when you take off the cornea, instead of throwing it away, it would have been a very nice idea to try to biobank this tissue and try to see whether there is a nice way or there is a strategy to transplant them. We know that is particularly difficult, but I mean, this is what people in our field are trying to do. In the meanwhile, because this tissue, they are taken from the donor with a very short post-mortem time, basically they seem to be very good for gene expression studies, and we are collaborating with many academic institutions trying to look at the retinal genes that are upregulated or are downregulated, for example, in dystrophies or in retinal disorders. We also have probably you see at the picture, sorry. We also have a Gmp Cell Factory for advanced therapy medicinal products, and it is basically a cell factory where we cultivate corneal epithelial cells for clinical applications, and it is authorized by our National Medicine Agency. And basically what we do is we have the possibility to cryopreserve these cells, mainly corneal epithelial cells, but we also have cryopreserved conjunctival cells, corneal endothelial cells, and retinal pigment epithelial cells, both for research purposes, but also for clinical application. Because we are working in GMP environments, it is possible for us to biobank these cells and provide them for clinical application, if they are needed. So this is also another important feature that we have. And as I was saying earlier on in one of my previous slides, is basically we don't only get healthy tissues from subjects, from the donors, but we also have the possibility to access diseased tissues and diseased cells from collaborating with surgeons that during their surgeries they provide to us tissues from people with, for example, genetic disorders. Like, for example, here I've put a couple of papers that we recently published where we are collaborating with a surgeon in Italy. He provides to us tissues from patients with Fuchs endothelial corneal dystrophy, which is a genetic disorder. And so we are trying to analyze the cells and see what the problems in these patients are. Or we also got access a few years ago to tissues from patients with the EEC syndrome, a genetic disorder affecting the P63 gene, and we managed to get both tissues and cells, and we try to see whether there is a gene therapy type of approach to correct this disease. On top of this biobank type of activity, so banking samples, we are also involved in sort of registries, so in I would call them banking information. So we have our own registry, of course, because we have to basically to trace where the corneas comes from, which donor, and where it has been transplanted. But we've also collaborated, and we are still collaborating with in European networks for having registering like this one, the CCTR, so which is the European Corneal and Cell Transplantation Registry, which has the aim to build a European web-based registry to assess and verify the safety, quality, and efficacy of corneal transplantation. So on top of banking samples, we also think that it's important to be part of these registries, so banking information, because they give you the possibility to access a lot of data and to do very interesting study, retrospective studies. So I believe that it was important for us, for our iBank, to be part of this winter school, because I think that the next step forward for our iBank is to become part of a more structured corneal tissue repository network, in the same way that we've heard you in Canada are doing, in the same way as the Americans, the American iBank Association is doing, where the iBanks basically they put on websites the tissue that they have, providing to scientists and clinicians the possibility to work together and to create new strategy and to create new work and new knowledge, of course. So I think it's very important for us to be part of this meeting today, because it would really be important to become part of one of these more organized, more structured network of biobanks. And again, these are our details if you want to contact us, so the website or LinkedIn. And with this one, I think I'm done with my presentation.
Thank you very much, Dr. Ferrari. Very interesting example of research collaboration and also clinical application, I would say, on a disease-specific biobank. So thank you for that. I will now invite Dr. Isabelle Fortier, Assistant Professor at the Department of Medicine at McGill University, to give her a 10-minute overview.
So thanks. Do you see it's not full screen? Huh?
No, not for now.
Is it full screen now?
It's in presenter mode, Isabelle.
Yeah, okay.
No, but we see your next slide.
Oh, okay. Sorry about that. So we did a practice, but it was not. Sorry about that. Okay, it will be. Let's have it.
Georgia can also show the presentation if it.
Yes.
Yeah. Right. Okay. Are you able to see even if it's not big?
Yes,
Yeah, I think so.
Screen. At the bottom of your screen, there's a.
Yeah, but if I do that, it's not working.
Oh.
Let's try that. Sorry.
No worry. If you want, I'm just a minute. I can share it for you. If you prefer, as you prefer.
Okay. I think now it will be okay.
We can see it.
Oh, yes.
Yeah.
Okay. Okay. Sorry about that. So I'm really happy to be here, in fact, but I have to mention that I'm coming from a completely different world. I'm an epidemiologist working at the Research Institute of the McGill University Health Center in Montreal. But in my world, a biobank can be a place where you put samples from different sources. But in my world, the biobank is more an epidemiological studies in which you are collecting different type of informations, including biosample, like UK Biobank is a biobank, or EPIC. In Canada, it can be Cartagena or Canadian Longitudinal Study on Aging. So it's, in my world, a collection of information that is broader than the information just collected around or for the biosamples. So in that world, there is a shift in the way we are conducting research. Each cohort or each source or each study is collecting a list of information and biosample, but often it's not the number of participants participating to one study is not large enough to allow some of the analysis we want to do. So, for example, interaction between genes and the environment or things like that. So we need to pull together different studies, the data from different studies, to be able to have sample size that is large enough to do the analysis we want to do. Or we can as well want to compare. If you compare what is collected by the sample from UK Biobank with the one from a study in Canada, it can be interesting just to compare, not necessarily to pull information, but as well to optimize the use of the data that are collected by these studies that often are used by the investigators, but it could be of interest to reuse in different settings the data that were collected. So it's a kind of, you probably heard about the FAIR. Everybody wants to be fair, so have data that are fungible, accessible, interoperable, and reusable. But when you're working with sample, you have the sample. But when you're working with all those other type of information, that's what you have to deal with. If you want to co-analyze data collected in UK and in Canada, you will have that reality where the studies are asking the questions to the participant or collecting measures that are different. In that example, you have the different questions that are asked for the job. So one study will ask, do you currently have a job or do any unpaid work outside your home? The other one, what is your job title? The other one, what is your current employment status? How do you compare that? It's very difficult. Or you can have, I don't know, anxiety scales or scales measuring physical activities that do not have the same construct behind. So it can be particularly difficult to co-analyze or to combine these information. But even for weight, okay, depending on the question you are asking, the weight, we know that the weight measured is not the same as the weight reported by the participant, because the weight reported by the participant will be more biased. Same for the height. So in some instances, it can be okay for you to pull together or to compare weight measured in one country to the weight reported by the participant in another country, but not necessarily the same. For me, it can be very important to just compare apples with apples, so just have the weight measure everywhere. So it depends. It depends on the research question you're addressing, what you can do with the data to compare them. Another complexity as well, particularly with the cohort studies, is that the information is generally not collected at the same time. So you can have, I don't know, a blood sample and the level of physical activity and weight measure at exactly the same time for one study, but measure with 2 years of difference for another study. So the alignment of the collection can be a challenge as well if we want to compare or pull data across studies. So we have to deal with diversity, we have to deal with complexity that are specific to each type of information. Again, if you have samples that are banked the same way at different places, it can be relatively easy to compare, but not necessarily even for the sample if the technique used to extract the information, the different biochemical measures from the sample are not exactly the same, the challenges can be there as well. So it depends really on the different type of information, the challenge we have to deal with. Another layer of problem is the capacity or willingness or not to share individual participant data. In some context, you will not be able to share at all the individual participant data. You will not be able to send a subset of data to someone outside of your institution. So in that case, the data from the participant need to stay behind the firewall of each institution. So on one extreme, you can, extract your data set and send it, let's say, one study in France, one in Italy, one in UK, and one in Canada. All the data are sent to the Netherlands, where the investigator is pulling the data and co-analyzing the data. But it's rarely possible. And more, we have to invent tools, invent ways to support federated analysis of the data. And Data Shield is one example of an approach that is useful to co-analyze data in different places, but without sharing the information. But you have other as well tools that are now available to support such situation. So what my group is doing, Maelstrom Research, is that we are developing tools, tools being methodological guideline, but as well software to help the cataloging, management, and harmonization and co-analysis of the data across different studies, generally cohort, but not necessarily only cohort, population-based cohort. We are as well, and I will tell you a little bit more in a minute, developing a central study catalog to help identify studies of interest and reuse the information that is already collected. And as well, we are supporting different national and international initiatives that want to construct the data infrastructure, allowing to co-analyze the data across different institutions or different countries. So currently the catalog, the Maelstrom catalog, like I mentioned, it serves to identify studies of interest and explore the harmonization potential. So for example, I'm interested in the level of cortisol and stress and of the mother after pregnancy, I don't know. I will be able to go on the catalog and identify the studies that are collecting the information of interest. So I will be able to contact the PI of the study and ask for access to the data and the sample collected by that given study. But I can as well explore if two different studies are collecting information that are similar enough for me to be able to combine the data across those 2, 3, or 5 studies. Currently, 23 networks, European and Canadian, and more international as well, including countries everywhere, are documenting their partner studies on the catalog. So 300 studies are documented there. And the variables collected by these studies are also included in the catalog. So really rapidly, in the catalog, you have comprehensive information about the description of the population, how the participants are, what are the selection criteria for the participant, the number of participants, obviously, but as well some paper that could be of interest to better understand the study content, and the contact information for the person responsible for the study to be able to ask for access to data or sample. But we also have the description of each data collection event. In that case, you have a study where you have mothers, fathers, and babies. So each line that you see is for one given, the different color is the different subpopulation, but each line is the description of the information collected and sample collected at that time point. So you can easily see when the samples are collected and when the information is collected. What we do in addition is that we take each variable collected at each time point and classify the variable in different domains. So here for the lifestyle and behaviors, it's that variable is about tobacco intake, that variable is about nutrition, that variable, etc. So we have 3 person full-time classifying the variables. It's long, but the fantastic thing is that when it's included in the catalog, you can easily search for information. So, or you are searching for design of the studies, or you are searching for a specific topic of interest. And that gives you, when you are searching, the following results. So you can, here what you have is the different studies and the different selection. It could have been the given biosample type of measure as well, or specific disease. But here you see, for example, that study are collecting 7 information about marital status, etc. And when the information, that's as well due to the when information is collected at each different data collection event. And it gives you as well the specific categories or format, the information about exactly the variable collected. That is helping, like I mentioned, to explore if the study have the information you're looking for, but as well it is helping to explore the potential to co-analyze data or how to co-analyze data across the different studies. So here you have an example of an information about ethnicity asked in different mother and child studies in Canada. And as you see, the subcategories of ethnicities differ from one study to the other. So here in 3D, they are asking if the participants have Aboriginal, Arab, West Asian, Latin American origin. And you see the differences in the different categories that are used for each of the studies. So looking at that information helped to see how we could combine the data across the different studies. So in that case, what the investigator did for that given variable is that they took all the one in green and set other for all the others. So there are different ways to harmonize or to process that information under the same format. But that was the way they did for that given study. So, like you just saw, there is a lot of heterogeneity when we are talking about population-based cohort and the information collected across the different cohorts. So when you go retrospectively and you take the information collected already, it's not the nightmare, but it's, let's say, challenging to combine the information across the studies. So in an ideal world, you would go before the data is collected and have common standards that are collected everywhere to afterward, in 5 years, 10 years, make sure that you can share apples with apples. And so you have example of, I think LT is a good example of a project where a group in Canada, a group in India, in South Africa and in China sat together. It's a project on mother and child. So they are recruiting mother before initiation of the pregnancy and wants to investigate obesity and different chronic diseases of the child through life. So they sat before starting and agreed on a list of common data elements that will be the same across all of the studies. Obviously, for information like income, it was not possible to collect information exactly the same in Canada and in India because the indicator of income differ across the different countries. But the researchers were able to agree on elements that can be transformed. And thus, in 5 years, 10 years, they will have a collaboration that can lead to a very strong ability to work together and collaborate. CAMPAT is another example where we work with different cohorts across Canada and generated over 3,000 core variables that are common across the different studies. And it's a total of 300 participants. So each study is unique and have additional data. So for example, Cartagena have more information about anxiety, nutrition, and work history than others, but they all share a common list of core information that can be pulled together and co-analyzed. And something that I should mention as well is the data structure behind that. In that case, you have some data for the study in Quebec that stay in Quebec, and the data for the other provinces that what naturally are owned by each of the study, but that can be pulled on a common server. So the complexity of the data infrastructure can be dealt with using different software tools and secure data infrastructure. Last thing that we are doing is that we are developing standards for documenting the information and providing access to the information that is harmonized across different studies. We develop our R package to help the process, but as well specific standard, because let's take the example of the weight that we had. It's not just to say we have weight and you have the list of weight that you can pull together. You need to understand how weight was generated to be able to make sure that the harmonized data that is provided is the one that you need. So thanks. That's the very fast overview of the activities of the Maelstrom team. Thank you.
Thank you, Dr. Fortier. I think also as research is really moving into the big data era and AI application, I think assembling with your tools a large harmonized data set will be certainly the future of creating those large data sets. So thank you for this overview. And I really like also the catalog framework to showcase different cores that are available for research. So thank you for that also. Let's move to the next presenter, Dr. Dileta Krojan. I'm sorry about the pronunciation.
No, do not worry, perfect.
Okay, she's the director of the Treviso Tissue Bank Foundation and president of the European Association of Tissue Banks.
So thanks a lot. Good morning and good afternoon. I'm so sorry, but our foundation is a little different compared to the previous presentation. Can you see my screen?
Yes.
I hope so. Okay. We are very similar to Veneto, a bank foundation. So we are located in the north of Italy, in Treviso, is 20 kilometers far away from Venice. This is our new establishment. And we are non-for-profit organization. And of course, we, as a tissue bank, we deal with all phases related to the donation, the processing, the storage, and the distribution of the tissues. Of course, it means that we have a quality assurance system, certification, and many training courses to update our internal operator and also the surgeon and the hospital transplant network operators. The difference between Treviso and Venice is that we are a multi-tissue bank. It means that we retrieve, process, storage, and distribute all kinds of tissues except ocular tissues, that of course is managed by Veneto Bank Foundation, and the skin, that is managed by another tissue bank in Verona. So we deal with musculoskeletal tissues, for example, bones, tendons, sternal plate, cartilage, like meniscus, for example, and nerves, for example, reconstruction of peripheral nervous system. Cardiac tissues, this is a very important issue because, for example, we are always difficult to provide a suitable number of pulmonary valves for children and also aortic valves. We manage also vascular tissues, for example, aorta, thoracic aorta, abdominal aorta, saphenous vein, and iliac artery. And those kind of tissues are very useful, for example, in bypass surgery, or when you have to replace infection prosthesis. Amniotic membrane provided by placenta, and this is very important tissues because we use for example, big burn patients, or for example, for chronic ulcers. In our specific case, for example, for oncological patients, we can provide tissue on demand. It means that when there is a specific surgery, for example, a big replacement, sternal plate or a tibia in oncological children, the surgeons can ask us a specific tissues for this major graft. We have two different accreditations. So, of course, our competent authority at national level is National Transplant Center, and they came us for auditing. And we are also certified by ISO 91. As you can see in the European Compendium, we are allowed to procure, preserve, process, storage, and distribute all kinds of tissue that I already mentioned. Our area of retrieval and distribution are different because there is a specific law said if you have a tissue bank inside a board region, you have to deal with this bank. But if you don't have a bank inside the region, you can make a specific agreement with another tissue bank. So we are the referent bank of other 4 regions. And like Veneto Bank Foundation, we can distribute in Europe and, if necessary, also outside Europe. Those are our numbers. So we deal with multi-tissue donors. This means deceased donor, and usually we arrive to have 200 donors per year, or living donors that, of course, they are more than deceased donor, and usually we have more than 1,000 donation. From multi-tissue donors, we can retrieve all kinds of tissue that I already show you. From living donors, we can obtain femoral head, for example, after a coxarthrosis, saphenous veins, placentas from cesarean delivery, and the cranial opercular, of course, only for autologous use. Again, those are the tissue that we are authorized to manage with, and recently we are authorized also to storage adipose tissues for autologous use, nerves and tracheal tissues. Again, those are our activities. So we have our own equipe to perform the retrieval, and they reach the hospitals where there is the death of the donor. So we can we have our own big car, and we reach also the middle of Italy 7 days on duty. And after the tissue came in our tissue establishment, and we start the processing in our laboratory according to GMP parameters. And we distribute again with our dedicated courier throughout Italy and in Europe. Those are our donors, so hair-beating donors that usually donate also organs, and our retrieval usually follow the organ donation. And non-hair-beating donors, so cardiocirculatory arrest, and of course this kind of donors cannot donate organs, but they can donate tissues, and living donors. Those are the technician while processing, because in Italy, if there is no a terminal sterilization, you have to process the tissues in A condition. So it means 0 bacteria or fungi circulating during the processing. So for this reason, they are were like this. Those are some photos of our laboratories. And those are some just photos to give you an example of our processing system. We have a 3 kind of preservation. So we cryopreserve for example, cardiovascular tissues. It means in nitrogen, and the tissues, the expiration date of the tissues is 5 years. For bones, usually we store at -80 dry. And also we have a freeze dryer to provide tissues that can be handled easily thanks to the possibility to store them at room temperature. When the surgeon, even in the night for emergency, needs a tissue, they just call us, and we reach all the hospital where the tissues are needed. Those are distribution. So we distribute more than 6,000 tissues per year. The majority is composed by musculoskeletal tissues, because of course you can obtain many types from one donor. Compare, for example, of cardiac tissues that we have a long waiting list for them. And so this is the connection between the tissue bank and the biobanking. As already Stefano mentioned, when a tissue is not suitable for transplantation, we can keep these tissues for validation, for training, and for this reason, in our tissue bank, we have also a research center that are on duty. We have a specific equipe to validate new kind of tissues, study a new type of preservation for specific tissues, or we study also some damaged tissues, as also Veneto A Bank Foundation perform. So I hope to be in time, and thank you everybody. This is our equipe, and thank you a lot. And I hope that the discussion will be interesting, but I'm sure.
Thank you so much, Dr. Trojan. I think that was a very comprehensive overview on your biobanking activities. The last presenter today is Dr. Bertrand Woodsie. He's an assistant professor at the Department of Medicine at the University of Montreal. So do you want to share your screen?
Do you see my screen?
Yes, but you have the wrong name under your.
Yeah, I don't know what's wrong, but this
That's
is my real
fine.
name. Okay.
Yes.
Yes, hello. So thank you for the invitation. Bertrand Woodsie, medical oncologist, clinician scientist. And today I'm going to shift gear and we're going to talk about feces and microbiome biobanking. So I can tell you it has been a struggle because I train in Toronto, then in Paris, and I've made home in Montreal in 2018 at the CRCHUM with Anne-Marie Mesmasson. And in 2015, a paper in Nature really showed that it was a struggle to collect microbiome feces of cancer patients at Gustave Roussy. However, over the last 6 years, we had a dramatic change, and in Anne Hahn's paper published last month in the Hallmarks of Cancer, the microbiome is now part of it. So in 2018, when I joined the CHUM, with the support of the Institut du Cancer, I became the director of the non-small cell lung cancer biobank, and we have now enrolled more than 350 patients. It's nice because it's an interdisciplinary team. We have now a project with radio-oncologists, thoracic oncologists, and we have expanded this network around the province in Quebec and with international partners. So what we do, every patient that comes to our institution with non-small cell lung cancer treated with cancer immunotherapy, they get screened by Julie, our research nurse, to see if they are eligible to any clinical trials open in our institution. If they are not, and are treated with standard of care, Julie meets with the patients and perform a dietary survey in order to see what they eat. She spends with them approximately 1 hour to go over the 10 pages dietary survey. Moreover, we collect the tissue, the FFPE tumor from either EBUS or pathological surgical samples. Then patients get PBMC serum, feces collected before their first round of immunotherapy, after 2 at 3 months, and we prospectively enter clinical data, CT scan evaluated by RESIST, ORR, PFS, OS, as well as immune-related toxicity. Everything is done at the CRCHUM by Mera, our technician. We then subsequently joined the Oncobiome Consortium led in Paris by Laurent Zitvogel, and all our samples are entered religiously in ATSIM. And as I told you, this network expanded in Quebec, and we have now 5 different hospitals that are collaborating for a dedicated project. And this has led to a lot of nice paper, and these are excluding the microbiome paper that we perform with our collaborator, thanks to this biobank. And again, I did not put flags, but this is from Switzerland, this is the Quebec registry, this is the France, this is from France also. This is the American COVID-19 Lung Cancer Consortium. This is with nuclear medicine and interdisciplinary imaging radiology paper. So this biobank really positions us as having access to various clinical data, radiological, and sequencing data, as this paper from our RNA-seq data from non-small cell lung cancer. But let's just move to microbiome, and without going into much detail, and those that treat cancer, I know that you're aware that we've shown that patients that receive antibiotic die faster, like our murine experiments. And with the help of this biobank, we also have been able to work with 34 different PI, and we published a meta-analysis of 12,000 patients demonstrating the negative impact of the microbiome. However, how do we collect microbiome? So we have 2 different approaches. This is the one we use at our institution. This is the more time-consuming, where you need a full-time technician at the reception sites. So we ask the patient to do their feces in this bucket, to put an anaerobic bag, and then they cover the bucket, they leave them in the fridge, they bring it to the hospital, it stays in the fridge in the clinic every day at 5, we go and pick up the samples, we open the samples, we aliquot, and we freeze it at -80. We don't put any RNA-later or conservative agent in order to use this for culturomic, metagenomic, proteomic, as well as FMT experiment. However, for other sites without the technician or physician less keen to ask their team to scoop the feces, we have a kit where RNA-later is placed in these brown top tubes. Patients scoop their feces in the RNA-later, close the tube, and send it by mail, and we receive it every day. And in the RNA-later at room temperature, feces can stay up to 1 week. With respect to the dietary survey, this is our news story, the impact of diet on immune response. And we modify FFQ, Canadian Nutrition Guideline Dietary Survey. And with respect to analysis of the microbiome, of course, we're shifting to a multi-omic approach. So we developed, thanks to the CRCHUM, multi-omic approach. So we have anaerobic chamber, MALDI-TOF for culturomics, Spark for OD measurements, and a room dedicated to cultivate, isolate bacteria. And we also have a brand new germ-free facility where we test feces combination with prebiotic or probiotic. And it's nice because we have been able to discover our first bacteria, Alistipes montrealensis, and the paper got accepted last week. But we also now have a biobank of bacteria because we have stored more than 350 bacteria that are very useful for collaborators at our institution or every elsewhere where people want to have access to these bacteria. And lastly, we recently published with the Oncobiome Consortium a validation paper demonstrating what we published back in 2018, that Akkermansia muciniphylla was enriched in patients with non-small cell lung cancer. And this large biobanking, multi-centric, 15 centers, 338 patients when enrolled. For Anne-Marie, 52 were coming from the CHUM. And you see there's a 10% difference in objective response rate. Patients with Akkermansia live longer. And at the CHUM, we perform RNA-seq tumor sequencing of the matching tumor when we had the feces. And we nicely show a correlation between the presence of Akkermansia muciniphylla in their feces and a more inflamed tumor microenvironment with upregulation of CXL10, CCR5, granzyme. So this is the first time we show that the microbiome could potentially dictate tumor microenvironment, immune contexture. And without going into much details, we're now not only collecting samples to predict, but we're also collecting and modifying the microbiome of our cancer patients. We have two clinical trials that are open. One is with fecal microbiota transplantation in melanoma and non-small cell lung cancer. And another one is going to open at the end of March with a prebiotic Camus recently published in Cancer Discovery. And by the end of the year, we're also hoping to have an Akkermansia muciniphylla trial in Montreal. And these trials require a lot of biobanking because we collect feces, stool, urine, serum at several time points. And we have a dedicated team to thoroughly collect, store, and share the sample and the data with our collaborators. And moving forward, I think that a multi-omic approach and strategy and multi-biobanking samples are needed and will be integrated. So we just finished a grant for Terry Fox, the Marathon of Hope, where 100 patients with non-small cell lung cancer NGS will be performed. We already have the matching fecal samples, we have the PBMCs, we have the serum that are analyzed, but the novelties, we also have radiomics, and we joined force with Imagia, which is an AI company, and we're trying to integrate everything together in order to define a robust clinical outcome. I just want to put a side note that, there's a lot of AI. People say AI, but I can tell you that AI is challenging. They will take a lot of time, and it's not that easy. And I know it's important to write AI in grants, but I have now 2 years of experience in AI, and there's a lot of bottlenecks, limitation, and things that need to improve on the company side and on the institution side. In conclusion, this is the team who's working day and night despite COVID to help us collect these samples and share the data and results with our collaborators. Thank you very much.
Thank you, Dr. Woodsie. That was a very interesting new field of research in cancer. I think microbiome research is becoming really promising new ideas for optimizing cancer treatment. So thank you for that. I think we are finished with the first part of the meeting. So we have heard from very different perspectives in biobanking. We have a 10-minute break scheduled, and we are a bit behind, but I suggest that we take a 10-minute bio break and that we all come back for the discussion session that will be moderated by Dr. Miss Masson. So the time on my computer is 10:35 in Quebec. So let's reconvene at 10:45 in 10 minutes. Is that okay with everybody, Georgia?
Yes, of course.
Yep.
Yes.
Okay.
Thank you.
See you later.
Thank you. I think almost everybody is back.
I think so.
Great.
So Stephanie, do you want me to
Yes.
start the discussion?
Yes.
So I first want to thank all of the speakers for presenting. I think what you've highlighted, especially for those who are following this as a school, look at the complexity of biobanking, the different viewpoints of biobanking, the need for quality control in biobanking. So thank you for your presentations. And we really saw, I think, in the presentations, two types of biobanking. Biobanking really for clinical purposes, so to be able to reuse those tissues for patients, and the type of quality control you need in such an organization. But we also heard about research biobanking, so the ability to provide samples to researchers to conduct health research. Maybe I can start on a point of governance, which which struck me, is that certainly in Canada, the informed consent of the patient at the time of donation for the purpose of research and what that consent contains is very important to be able to allow us to use samples for example, genomic studies or providing them to large biobanks. And I was struck that for many of the clinical biobanks, you have a very Dr. Pigatzi, for example, has a very rich retrospective biobank. So perhaps you can talk to us, has there been any challenges related to consent, and especially when you consent to providing to other countries too, because I imagine that perhaps Europe is not unlike Canada. We have provincial laws that govern it, and so we often have challenges when we have collaborations of actually putting samples together from multiple jurisdictions and bringing that data together with consent.
Okay, so as I mentioned, we are part of this Italian association, which is IELP. All samples we received are from patients that have been enrolled in a clinical trial, in an IELP clinical trial. These that patients, so parents or legal guardian, sign an informed consent where they allow the patient to be enrolled to a clinical trial, and together with this consent, they also had signed that all samples that clinicians will take from the child will be used for diagnostics or for several experiments that will help in clinical decision, and all the material that is that remained is being stored in a biobank that will be at service of scientists for research or for projects that will improve knowledge of the disease or improve in health about what we can reach with science. So we received samples with this consent signed, and we stored it. We have when we managed to have a collaboration, and so someone are asking for samples from our biobank, we contacted the Italian coordinator of each pathology. So I'm working mostly in acute myeloid leukemia, for example. So I received a proposal for a project for increasing a cohort of AML. I talked to the Italian coordinator. We are a small group for AML. We are 5-6 persons from all the Italy, and we talk about the project. And we, if we think that the project is and that samples may be used for this project, we agree with this proposal, and I take contact with the PI of the proposal Italy, and we decided how many samples, and then we sent the samples by signing an MTA, so a material transfer agreement. They received samples, and then, when necessary, we share also the biological and clinical data, since most of our studies are for clinicians. So we also will share the age, the sex, and the drug, which is the trial where the patient is enrolled, because it's very important to know which drugs have been used with the sample, and all the info they would like to have for that are included in the proposal we have seen and we have also accepted. So this is it's very easy way to work, because if the project is good and we agree to participate, material is sending only via MTA agreement.
Thank you for that information. Does anybody want to add to that?
Yes, if I may, because in some cases we are some similarities to Martina, but for example, in our case, we have some critical point for deceased donor. These issues came up in the last month because from legally point of view, there is this topic. So the family can sign the consent also for research, because of course they, from legally point of view, the family can sign the consent to donate the tissues for transplantation. And as I mentioned, we can keep for research validation the tissue if they are not suitable for transplantation. But now this issue came up in the last month regarding the suitability of this consent from the family, so from legally point of view. So we are discussing with this. Whereas from living donation, usually we have or a specific consent for the clinical trial, like Martina said, or we ask a very basic consent within the donation, and we provide the information to the donor that if the tissue is not suitable, they can help to improve the knowledge in the transplantation studies. So we are blinded in this area. Otherwise, we should follow Martina ways. So we have this little difference in our work.
I forgot an important thing, that all IAEA clinical trials are reporting all the steps that patients will undergo, so clinical steps, but also there is a part which is called Ancillary Projects, where we describe briefly the projects that will be done with the material for this research. So parents can read in the consent for what they are saying. Yes, we allow the use of the residual material for kind of projects that are where we describe, for example, gene expression, proteomics analysis, or in vivo PDX development. So parents read this long information sheet and then decide to say yes, we allow for research or not. So all trials are included in the all trials. All our projects are included, so they can see and for that for the projects we have already decided when we write the trial.
So maybe we can change topics and talk a little bit about data. Data is often a challenge. How do you share the data? How do you collect the data? You've said that you collect it from clinical trials, and we collect it from hospital records. We've talked about the difficulty of harmonizing the data and whether AI is really a solution or data lakes are a solution. So do people want to maybe express themselves on the difficulty of actually acquiring certain types of data and sharing it across networks, and whether the new privacy laws that we're seeing have an effect on our ability to share? And I see that Bertrand has his hand raised.
Yes, yes. So we at the SHUM, we have the chance to have a data lake, AI company, but unfortunately, I have to admit that because we use several programs and for the pathology, for the pharmacy, for the lab, for the physical exam, and the notes are either dictated or still written on paper, it has been very challenging / impossible to fully use the data lake, and we're still using old school Excel spreadsheets. For the clinical data, we have a REDCap, of course. When we exchange with collaborators for a smaller project, we tend to send our Excel template with the codes and the dictionary. 1 is female, 0 male, ECOG 1, 2, 3, 4. So everything is standardized. But I think we were much behind, at least in Canada, in terms of using all the possible and novel strategies to collect new data. And I don't know what the situation in Italy. We work a lot with Nicola Segata from Trento University, and we're with the Microbiome data. And unfortunately, some are in REDCap, some are on Excel. But I think we were years before using data lakes or AI to get clinical data in Canada, unfortunately, despite the SHUM being #1 connected hospital in North America.
So I see Isabelle has raised her hand.
Yeah, but I would say yes. You know what you're talking about. I think that it's you're right, but it's more complicated than that. You have the format, so a format in SAS and it's PSS in Excel and etc. You have as well the way the information is collected, male, female, perhaps it means the same thing, but smoker, non-smoker, perhaps even if it's smoker, non-smoker, it doesn't mean the same thing depending on how the information is collected. So that is adding challenges as well. So at each center, you need, like you mentioned, to put together sources, so to link different sources of information, and at each linkage, there are difficulties for the completeness and quality of the information that is integrated. But in addition, when you compare across different places, all the documentation, and like you mentioned earlier, Martina, the documentation of how the information is collected is so important, because sometimes it's not possible to compare or to pull together the information across different centers, or it's not relevant to use some information if it is not as good as you're looking for. And it's okay to say, no, I will not use that source of information. It's better to say that than to try to build something that is not as good than that, where you will work for 1 year to try to build your data set, and at the end you will just have one not very good paper that is published using that data set. So you should go for quality and really strong standardization, or make sure that you have exactly the same meaning when you're comparing or using data.
I can add something. We are working together with the Cineca. Cineca is an Italian consortium, but also an European consortium, which provide high performance computing systems. So we have a system for all Italian centers. So each center registers the patients into this computing system. For each sample, a system assign a number, which is an impersonal number. And from this point, everything we are doing for patients will be registered into this system. So when you would like to have information about patients, you can digit the number. You can have all the information that is being scheduled and are all the same for all patients. So this is a very, also the reports or the experiments the experiments we do and results are put into this system. So each center can see the reports that include in the system when we perform the analysis. And at the end, Cineca is also able to perform queries, and they extract each variables you would like to have. And so everything is very systematic. It is controlled by a consortium that is doing that for several universities that collaborates with the industries and also with the public Ministry of Health and of instruction. So it's a very important tool that we have, and this means that all the information is recorded in the same way, and so we can work with data together and do easily queries for each question you have, each clinical or biological question you have. And this is also this could be also able to extrapolate or to put together different data sets in order they can talk together. So if we would like to merge my data sets and your data sets, the systems would have the ability or the possibility to do that. And here, as I hope in Italy, we are doing together with Chicago University, who has a group that is working on that, in creating dictionaries for each disease, and trying to put several data sets from the biggest centers around the world all together to have a master data set where you can put your records of the patients, but then you can also use these data sets if you would like to make a bigger and bigger analysis. So it's a very complicated system, but it seems to work very well. And this is also backed in the quality certification we have for the ISO 901, but specifically for the accreditation, so the ISO 51809. And everything is okay with the Europe and also with US, since we are working also with companies. So for example, clinical studies for the experimentation of a drug, we work with pharma, so Novartis, Pfizer, and for working with this kind of stuff, you need to be very to follow strictly rules, and this system is able to give us this opportunity and the ability to do that.
Isabel, you want to respond, and then maybe I will help.
Yeah. I totally agree, but that's true when you're talking about diagnosis or sample, but when you're working in the world of population-based cohort, you have to deal with one cohort, let's say UK Biobank, that measure physical activity with one given scale, and another study with another scale. So the differences in the way the information is collected is very challenging. So that's why if you go prospectively and the different cohorts agree to collect the same, to use the same, I don't know, anxiety scale across all the countries, that's fine. But if not, the challenge of pulling together information cannot just be enter the information in that given environment. You need to use additional techniques and methods to be able to process the information under the same format and co-analyze data. So when you're talking about gene-environment interaction or more epidemiological type of information, the challenges are a little bit different.
However, I think we should learn from the Italian and the European, and I had the pleasure to spend 3 years at Gustave Roussy, and I work a lot with Italy, is that they have network and consortium that work together, they share data, and they are very powerful through their connection of the data. And in France, you know, they have the TIMOM group, so all the rare cancer all enter prospectively in a data set. And in Quebec, with in vivo, we had the hope to do this one day, but I don't know where this has been leading to. And I think this is really what the Europeans are doing extremely well, is to collaborate together, work together, share Excel spreadsheet or RedCap. And we really need to learn this in Quebec and in Canada.
Yep, I would like to underline that before doing databases and data sets, we previously decided dictionary. So we have to harmonize our variables in order to then to be more easy to merge together. So yes, if you have different variables than ours, because we're talking about cancer, but also for cancers, because in Italy we are talking about complete remission in a way, and the US complete remission in another way, and also in Germany and so on. So before doing to merge data, we decided a dictionary that will be the same for all the groups. Only then we can merge data sets. So you need to speak the same language before doing this merge of data, and this is very important. So the biggest work is doing a dictionary for each disease, and then talk everybody the same language about this disease. So this is very important.
Maybe I can ask a question to you, because you mentioned that you work with pharma, and certainly one of the challenges in clinical trials is first, you know, during the randomization process, you may not know who's on drugs. And there are certain trials that even after closure, at least in the North American experience, pharma does not release who was on what arm of the study. So do you ever experience that problem also? And how do you deal with it?
No, not specifically this problem. What we do with pharma is give a service that is selection of the samples and so of the patients that will be enrolled in a specific clinical trial. So we perform, for example, we are talking about biological drugs, so drugs that are targeting a mutation. So what we do is performing the mutation, and so allowing and saying, yes, this patient could be enrolled in this clinical trial. We work at basis. Probably it's not, you are talking about a step after, and most of the new drugs that we are testing are not the first line drugs. So this also randomization is more used for the first line drugs, and the new clinical trials and experimental phase 1, phase 2, and phase 3 trials regards patients after relapse, after the first, the second relapse, and rarely we are going to randomize drugs in that point.
I don't know if anybody else wants to comment on that, or.
Well, no, I just want to comment, Anne-Marie, I just want to comment something about, you were saying, and somebody else said about working with pharmaceutical companies and industries. And because all of us are involved in biobanking activities. I remember we had a few months ago a meeting with our Italian National Transplant Service, all the people working in the transplantation, and we were discussing what Diletta was saying earlier on about having the tissues not suitable for transplantation and therefore available for research, and therefore having the consent form signed for research. And everybody, of course, was happy about that. I mean, apart from what Diletta was saying, that, I mean, we have now some issues about using these tissues for research, but having said that, everybody is very much happy about using tissues for research or whatever we take from surgeries to be used for research purposes. But then when it comes down to collaborating with pharmaceutical companies and industries, I mean, people were very much in a sort of a dilemma, because we are asking the donors, or we are asking patients for their tissues and their cells, which are fine if then we work with academic, we work with clinician to advance with knowledge. But when it is working with industries that of course try to make money out of it with information that we give to them with the tissues, I mean, people are less favorable about using these tissues for this type of studies. So what is your perception about that? I mean, is it an experience that you also have in Canada, or like Martina in her studies, that basically when we have the tissues, when we have the information, when we have the registries, one thing is to work with non-profit institutions or public institutions, while different is if we work with companies?
So I can tell you what our research center, the philosophy behind that, and it's that we work with companies when it's a collaboration. So in other words, we would not provide a company for which we don't have a project with, for which we don't have a study where we are asking the same question. If they just came to us and asked us for tissue, the policy at our research center is not to share tissues with industry. However, we also understand that if you want to get drugs to market, if you want to get new tests to market, you have to at some point collaborate with industry, because they're the ones who are going to be delivering the new knowledge into the clinic. And so in order to, we favor collaborations because there's a partnership of trust that establishes itself with the company. We have shared goals with them, and usually, you know, the terms of the collaboration are set out in the contract and the MTA. So that's pretty much the philosophy. And our consents, our consent clearly indicates that we can, if we choose to collaborate with industry. So they know that that's a potential when they donate, but that has not been a big problem.
So you inform the donors or the patient that whatever tissue or piece of information you get from them, it might be used for collaborations with companies, and that eventually, I mean, the companies might make, of course, I mean, money out of it, out of this piece of information, and they don't get anything back.
That's clearly stated in the consent.
Okay.
So in the actual consent form that we use for the network, I can't say that all consent forms are like those consent forms that don't identify industry. It makes it very difficult to share those samples with industry. So it has to be part of our general consent form.
And all your patients or family of donors are happy with that. I mean, I've never had a sort of backlash from this.
No, not presently. And I think one of the important things is that it's, so our bank is a general bank. It's very important to have a relationship with different patient groups, with patients themselves. So in Canada, we've developed over the last couple of years this whole notion of the patient partner. So we have patient partners that consult with us at the Biobank, look at the kind of studies that we approve. So they're a voice for the patients in terms of what gets approved or not. We also, on our websites, declare which studies we are doing with the samples. And so we have as part of because it's a general consent, we cannot say your tissue will be used for study X, because we don't know in 5 years what that study is going to be. So our responsibility and how we deal with it in the network is that we have a website where we show which are the publications. We try to explain the research we do. We show where the projects that are being funded and have access to the tissue. So we try to make that information very open so that patients can see it and can comment on it. We've had very, very few patients retract consent over the years. It does happen, but it's quite rare. I think that having a good communication with your patient base is very important for that.
Thanks.
Yes, I agree. And if I can add something, in this moment, we are collaborating with pharma since we are doing a service is useful for the patient. So you explain to the parents that doing this analysis will be suitable for having a new drugs for the child. So this is another way, and we are not making money, and also the company is not making money because the company is giving the drugs by free, okay? Because this is a new drugs, so we are doing clinical testing and experimentation. This is a way where no one makes money, and also this is experimentation. And the second line of pharma and academia content will be using the samples and for example, we use primary samples to do patient-derived xenographs, so modeling of cancer into mice. There are consortia made up of academia and companies that are using the model to discover new drugs. So if a company, by using my PDX, will discover a drug that will be fantastic, they will make a lot of money, and you are right that in this kind of studies, pharma can make money by using samples that we are producing. You have to explain to the parents that in some cases, in these consortia, they are giving you money to do research. So if you give samples, you will have a piece of the fund that this big grant will have. So I will receive money for doing research, and parents need to know that they are doing this in a way that for the future, and you will have more knowledge, and in the future you can use new drugs only if we are able to do that. So it's like invest the samples of your child for future children that will be cured better than now. And this is what Anne-Marie said. So the best way to convince parents to sign this consent is explain what we are doing with the money that company will give us, and try to make understand that this is a virtuous process that need to go on also with the company's help, if necessary, if you want to participate, if you are involved in this kind of projects, European projects. There are two big consortia in Europe, ITCC. They are also initiative funded by the European Union, so there are a lot of money for doing that. But pharma are involved with academia, with their experts, and academia have a lot of samples and are doing models for us, because we are working on that. But if you want to share with the companies, you can have research money, so grants funded for going to the same results. So the objectives will be the same, mind of the company and of other European groups. For example, immunotherapy now, we have a lot of money for doing new cellular-based therapies. There are grants funded in this way. Yeah. It's not easy. It's very hard for parents understand this kind of stuff. You're right. It's much more difficult.
I saw that Renee had her hand up, but she seems to have disappeared from the meeting at the same time. So there she is.
Yeah, okay. Yeah, I got disconnected. I don't understand why. I just wanted to comment what you said, Anne-Marie. It's very similar from our side, where we collect not tissues or products from patient or from healthy donors, but we have the same philosophy. So if the material is to be used for research for a company, we really look into, will that benefit the health of the Quebec population? If it's the case, and we are willing to participate and provide that. And the informed consent really specifies that there's a company that is involved, and that the the participant will not have any benefits from that, except that it will contribute to improve the health of the population. But sometimes we refuse some projects because we just think that the company will make money out of it and not really benefit from the population. Mm-hmm. And the other thing is, for some line of products like the milk or the cord blood, we have a more generic consent. So you agree or not to have your material, if it's not suitable for transplantation or bank, you agree that it will be used for research, but we cannot specify which research precisely. And so we're doing the same. So we are listing the projects that are already approved on our website. And so the participant can just look at the project, and if they refuse to participate, so they can contact us to say, okay, this kind of project, I don't want you use my material for this kind of project, but for the other projects. So that's how we manage that. Yeah, because there is a lot of demand for our products from healthy donors to be used as controls, for example, in lots of studies, and and it really right now it's not optimal the way we can provide these samples, because we need to get back to the people, the participant, to get informed consent, for example, for blood products. So this is a big challenge for us.
So maybe we turn to a different topic. I noticed that several of you talked about the ISO, and it's completely understandable. Many of the biobanks we've heard of from the Italian groups happen to be also, clinical repositories, which have to, provide tissue to patients. So, but maybe you can speak about some of the smaller biobanks across Italy. Yourself the expense and the time it takes for ISO qualifications. Do all biobanks in Italy have some? We created the CEDARNet certification and program really with smaller biobanks in mind, by academic biobanks that are maybe very fit for purpose and are not big clinical providers of material. So can you speak to that side of biobanking in Italy?
So to my best of my knowledge, ISO is not mandatory, it's just recommended. And so I do not think that all, mainly smaller tissue bank, biobank has ISO. I think that is only recommended, not mandatory, but I do not really, I'm not sure 100% because we are certified by 20 years. So I think that is only recommended, not mandatory. But I have no direct experience of a biobank not qualified as ISO.
But ISO 9001 is a general ISO, but there is a more specific biobanking ISO now that has been released. And so have you also certified for the biobanking ISO?
No, we are going to. I know that in Italy it's very new, this ISO, specifically for biobanking. We ask to be accredited next year, but until now we are not.
And do you know that the money and the time it would take to certify for such an ISO?
No, I do not know, but as you mentioned, Anne-Marie, as we are a bank that provide tissues for transplantation, when we have this discussion with the inspector, they told us that usually the procedure that you have to establish, they are really similar to the procedure that we have already had, and usually is the level of that you have, for example, of the attention and risk assessment that you have for to provide the tissues for transplantation is higher usually than tissue that you provide for a research purpose. So they are very comfortable with this. The inspectors say, do not worry, it's not so time consuming for already established tissue bank. but really I didn't perform the really analysis until now. So maybe I'm just a dreamer.
Yeah, we are accrediting the labs and all the methods because there is a mandatory European rules about that if you would like to enroll patients into international trial. So you need to be an accredited laboratory if you would like to that your analysis and your reports would be recognized for enrolling patients in an international trial. And most of our trials now are international. This means that an Italian child with acute leukemia will be follow the same trial of a German, a French, or another person who is joining the same trial. For the biobank, since biobanking is used for research or for retrospective studies, the ISO is not mandatory for now, but will be from 2023 for being an accrediting lab, and being the accreditation of the biobank will also be asked for. I think this is a very time-consuming activity. The accreditation is very, very strict rules, and we have to do a lot of documents. And about the money, have to have a consultant, an external consultant who is able to do this kind of stuff for accreditation. It's a very it's an expertise that nobody has if you don't do this kind of job. And then you have to pay the visit. So for being accrediting, external people are coming to your lab and see if everything is okay. This of visit, the audit, as we called, need to be paid, and it depends on how big is your bank and how big is your lab, and how many days they will stay there for visit and for giving the final results. So it could be less or more expensive depending on how big is it the biobank.
And do you have a registry of biobanks within Italy? Like if you went to a university, would you know how many biobanks are in that university? One of the challenges we have is that, within an institution, it might know which biobanks it has, whether it's clinical or research biobanks. But as a country, we probably couldn't tell you.
I don't know if I understood well the question, but there is an Italian repository of all the biobanks of Italy, which is called BBM-RAI, which is an online website where you can look at the biobanks that are in Italy. And I can talk about for the pediatric cancers, we are the reference center. So there is only one biobank that is here in Padua that we are leading. So for tissues, diseases, and so on, there is a website where you can look at Italian biobanks.
And are all Italian biobanks necessarily registered there?
No.
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 Actually, I think you did very well, Martina, to raise this point about the BBM-RAI, because it was supposed to be the Italian branch of the European BBM-RAI network of biobanks, but I think it's on a voluntary basis. So if you decide to join the network, it's fine. Otherwise, like for example, up to 2021, we were part of the network, and now we are not. So it depends whether you want or not to be part.
The same for us.
I know that in Canada, there is the CDTRB, which is a Canadian network for research on transplantation and donation, that tries to regroup all the biobanks and give an access to all the information. But I think you have to be part of the network maybe to know about this initiative. I don't know if it's widespread around the country.
Yeah, well, CTINet was founded on oncology, so we have a better idea of what the oncology biobanks are. But I'd say even there we get surprises, where we think we know all the biobanks in our institution, and then we discover another one, you know, a couple of years later. So there's a challenge in registering all of these biobanks, and for even institutions knowing all the biobanks within. Maybe I could ask you a question too, because we talked about patient partners. I mentioned we do patient partners, and they're an important they're important colleagues in our work going forward. But it's a fairly new notion in Canada. You know, I mean, it's something that is gaining a lot of traction in the oncology setting. It's something that we've seen a lot. It's coming online with other disease sites now. Do you have patient partners, and is that a movement that you see in Italy, that they're your partners in these types of studies?
When you say
you mean
partners, do you mean patients associations?
No, well, there are patient associations, but we're talking about actual patients where there's even we do some formal training with the patients so that they understand research, so that they understand the language of research, you know, because if you just throw a patient into a room with researchers without any knowledge, they're quickly overwhelmed by the conversation and don't follow. So, you know, we have a program of training of patients and then incorporating patients within research groups or within, biobanks, so that they can be a voice at the table when we have discussions.
No, I think no. We, from my side, we don't do that because we have children, so we cannot try to make children understand. And for parents, everything is based on the first talk with clinicians and parents at the time of diagnosis, and then all during the period that the children are in hospital, we continually talk with parents and try to explain, because only also during the follow-up and all the steps of treatments, we're monitoring minimal residual disease. So parents are continuously involved in this kind of discussion, trying to make them understand what we are saying. So, but we don't have a kind of staff or something organized to make them more conscious about that. But if they ask, we have also psychologists. So most of our psychologists are following family also for understanding, for having a communication, also for scientific part, helping us in make them understand them more and more, but not something organized.
Well, I just point out that in the pediatric world, we do have patient partners. As you say, they are the parents. They are often the parents of children that are deceased, but they continue to be involved in their network because they want to follow research, pediatric research, and they want to contribute back. So you can involve parents long-term, even from deceased children. But that's just, I was just asking about your experience, because that's the Canadian experience that we've seen now.
Yeah, it's fine. It's a good idea.
Yeah, well, we have something, I mean, probably similar to what you were saying, because, for example, normally when we receive a donation, a corneal donation, normally we write a letter to the family of the deceased person to basically thank the family for donating the cornea. Basically, this is what, and very often we do invite the family to our foundation. We do invite the family, and if the corneas have been used for research purposes, we try to explain to them, because at the beginning many family of donors were really unhappy if the corneas of their beloved basically were not used for transplantation, because of course, I mean, transplantation is felt like the person is still living with us in a way, but if it is used for research, basically, it was a sort of betraying the donation. But actually, so we decided to invite the family of the donors over to our foundation to show them what we do also in research, in what we do in research. And actually, this really helped the people to heal and also to understand that what we were doing was good, and the donation of the cornea of their beloved mother or father or son was good. If it was for transplantation, it was good today for one person. If it is research, it will be good tomorrow for many more people. So we do something like that. So inviting personally people, the family of the donors to foundation, explain to them what the research is, and we show to them our research labs. We let them talk to research scientists. So just to they feel we think that they feel better actually.
Yeah, I guess that works if you have a small number of people that you want to invite. It can't work on a very large base.
Yeah, of course.
One of the things we do with patient partners actually is they help us produce material that speaks to the patients, you know, in a language and answers questions that they have. So they're also a very good source of information of how to communicate with your donors. They often chide us on the language we use because it's either too scientific or it's not addressing actually what they want to know. So they can be very good sounding boards in that way.
Yeah.
What we are doing, and I don't know if it's a widespread practice, but we have a representative of product donors and recipient in our research ethics committee. And I have to say that they bring really interesting points regarding the use of material in the kind of project that are looked by the research committee in terms of, well, would a donor really accept that? Or so they are really participating into the decision, and I think it's a good addition to have on these kind of committees.
I did want to mention too, I think a number of you mentioned that the importance of training the next generation of scientists and biobankers and using the material that you've collected for training purposes. I think it's critically important. I know that in Europe, I don't know if in Italy, but there are actual, you know, graduate degrees now in biobanking so that people are professionals. It hasn't made its way yet to North America. It's still something the highly qualified personnel acquire the skills training on the job and not necessarily as a formal education. But I think that's a big difference between Canada and Italy too, maybe.
Well, I don't know about courses of the academia. I don't know, but probably there are some. I mean, I don't know everything about Europe, but I'm sure there are some university courses about donation and disaster stuff. I know that, for example, both the European Eye Bank Association, but most of that, the American Eye Bank Association, they have certified eye bank technicians courses. So you attend them and you become a certified eye bank technician. So that's for sure. I don't know about in the universities how many courses there are about this. But we do think, and I think that Diletta was saying this earlier on, that in Italy the law that on which is based the old donation, organ and tissue donation, basically states that it is crucial and it is fundamental for this activity, for the transplantation activity, for example. It is crucial and fundamental that the bankers, tissue bankers, and the surgeons are trained with the tissues. And so there is a lot of efforts, at least in Italy, but I know also in Europe, for like wet labs to go on, and so to train all these people. There are also companies. I mean, a few weeks ago, I went to visit a company in northern Italy, and basically they import bodies from the US, because in Italy is not very easy to do, but they import whole bodies from the US to do basically training for surgeon, to do training for orthopedic surgeons, and for neurosurgeons. So I mean, this is very important. I think in Italy and Europe, this is an activity that is going on very well.
Yes,
go ahead. Yeah.
No, sorry. I know because I've been involved in the last 5 years. A university in Spain, in Barcelona, they provide a master in tissue transplantation coordinator or something like that, and they are specialized. They have specific course both for retrieval part, processing, and also clinical application. And they involve the European tissue bank operators to also have a technical course. For example, we went to Barcelona University and we explain how we assess the suitability of the donor from external point of view, internal, and so on, and how we process technically the tissues. So as far as I know, this is one of the major master. I agree with Stefano. Otherwise, I do not know if there are any other specific training at university except this.
Isabel, maybe you can tell us about training in terms of data harmonization and data collection, because that seems to be an art in itself or science in itself.
Yeah, nobody is trained for that. We have to train, the only, person we can find are data analysts, but they will need to be trained by our team. So there is no course specific to the harmonization per se. And even, I'm teaching at the McGill some courses to clinician or to people involved in clinical research about just, be aware that zeros and ones, it's not, enough, that you need to, go for quality. And in your world, I know that's something it's your daily life. But for a lot of the, postdoc or fellow that are jumping in and have like 6 months to do a research, they will take the data that is there, but without necessarily putting enough time in exploring the data and making sure they understand the content of the data set before doing their, paper. So that is something I think that is extremely important, and we need to repeat and repeat and repeat again to talk about the quality of data we are working with, and even before thinking about harmonization, thinking about quality.
So if we, on the quality topic, we've talked a lot about ISO, but ISO is very much a standard that very much tracking. It's almost being able to be a detective sleuth and be able to go back in your records and identify who did what and where and how. But it doesn't have a lot of quality management around things like governance and ethics, and maybe some of the things that are perhaps more peripheral, like education. So is there any, is there some sort of, is there an equivalent in Italy of something like CEDARNet, or do you refer yourself to ISBER when you think about subjects like governance and QC, quality management? Or it doesn't come up much because you're very much clinical lab, so it's not. Martina, I think you're on mute.
Sorry. Yes, we are following academic, so university and hospital rules more or less, and trying to follow the ISO, since hospital asks for ISO accreditation, and so we follow this kind of rules. So government is giving rules to hospital and academia to us. So this is we are in contact with the governance or the government for this part. So I think this is how we manage this part.
Maybe I'll throw it open to you. Maybe a more open. Is there anything that you would like to discuss or bring up or question in terms of biobanking? Maybe I can ask a question to then about live biobanking. So when you have, are there any challenges in terms of, you know, the long, in your MTAs, when you transfer live material, about how long the investigators can keep it, for what purposes, or do you share the material freely, and how do you honor consent when things get passed around internationally?
I send samples when we agree with the project. The second step is work together to data, to results, the paper. So we have scientific aims, and so finally we would receive names, authorship in a manuscript. So this is our way. We are academia, so our collaborations are in writing, so manuscripts, or preparing preliminary results, participating to grants. This is what we have from this kind of shipment and sharing samples.
But if you, for example, if you publish a PDX, and then I were to write to you and say, can I have access to that PDX? I've now seen it in a publication. What is your policy then for sharing such a PDX?
And the policy is the same. So you ask me a PDX, you send me a synopsis of your project. So you ask the PDX for doing what, and then I ask to Italian, to IELP, to coordinator if they agree to the PDX with you, but you have to sign that you will use the PDX for research purposes and scientific purposes, not for making money or sending the material to others. You use with the aims you have written in the proposal, and with scientific aims, and nothing else.
Yeah, perhaps that's the easiest part of the MTA. What we found often is a challenge in the MTA is the intellectual property.
Yeah.
So what that's when the lawyers really start to sharpen their pencils, and you know, where we say, hey, if there's new discoveries, who owns the intellectual property, how is it shared? So is there a standard in Italy for sharing of intellectual property?
No. So intellectual property, if you have something new, you make a patent. So the intellectual property is of the university in my case, and if you would like to use my model, you have to buy, or I have to license the patent to you. And this is it is like for companies. If I make a patent of something and a company would like to use it, they will pay the university for the patent, and so buy the patent or have a license for 1, 2, 3 years of the patent. And this is a stuff that academia, so university, is working on, because if I made some discoveries with the patent, the intellectual property is mine as inventor, but the owner is the university. But if I have if I published scientific data, they are already global, so you can use them for scientific purposes. The intellectual property is already mine because I published this data before you, and in your paper you will write that I will be a co-author, or you, in the acknowledgements, you will say PDX have been kindly donated from Martina Pigatzi, University of Padua. More or less.
That's the easy intellectual property, because that's the intellectual property that you generated. But what happens if you give a PDX to somebody and they now generate new intellectual property?
It is not intellectual property as we mean, because for me, intellectual property is something that you have patent. You have a patent for that. This is an intellectual properties with a more official and benefits of money. The intellectual properties you I'm talking is about scientific discoveries and publications, which is not an intellectual property because I didn't patent before, so it's of the world. I do research, I try find new data and I publish them. So there is no intellectual properties because there is nothing new.
So if you share samples with a company or you share samples with another researcher and they make a whole new discovery and they have a patent, then
if you do that, we make a contract where we decide who is the owner of intellectual property. And we can say 50-50, 80, or this is another way of working. And if during a commercial a grant which is funded by an industry, so industrial grants need to have a contract, and in the contract we have to decide who is the intellectual properties owner. That since I am a university, I'm academic, I'm associate professor, University of Padua will sign the contract with the company, deciding in which percentage the intellectual property is of the academia or of the company. So everything is discussed, depends on the grants, on the discovery, which is the object of the proposal. But you have to sign a contract in this kind of stuff. Yes, you need to know and to sign and to decide before the intellectual properties. Yeah.
Okay, I think it's pretty much the same way as in Canadian universities. The universities are the owner of the intellectual properties.
Yeah, universities are always the owner.
Yeah,
a company can buy the patent, or they can license the patent for a few years. But university is doing this kind of stuff, yes, for us. And I think that in Italy is also rules that are available for all different academias. So the government, so the Ministry of Instructions probably is deciding how to govern this kind of stuff.
Maybe one last question, because I see we're running out of time, but to Bertrand Woodsie. I mean, you spoke to us about AI. We hear about AI, that it's going to be our savior and that it's going to help us go through all the data and solve all of our problems. Can you just perhaps you mentioned it in your talk that it's at least for now it's not the end all and be all. So perhaps elaborate a little bit on that theme.
Yes, so I think there is unfortunately a bit of a disconnection between people on the field that work and people who take decision or even invest money, and or at least in Canada, I don't know the situation in Italy, but in Canada, the government has invested millions and millions of money in AI company for research, and the relationships are challenging because there's a lot of competition, a lot of conflict of interest. Some institution belong to one company, the others, so you cannot work with a different institution. And there is these conflict of interest, I have to admit, are way different than the one we're used to see with big pharma, where the big pharma, they know the rules and people are used to these rules. Whereas the AI biotech seem to be a bit more sneaky, or this is one challenge. The second challenge is, of course, to give access to these AI platform to patient data, and this in Quebec has been a big flop and a major failure because we cannot give access to patients to AI groups, AI company, and we've struggled a lot. And then for the AI company, I think that they sell a lot of dream, and I just watched yesterday Elon Musk thing on AI, and he says he's going to revolutionize the world and they can do extraordinary things, but I would be very keen to see any AI PubMed paper published by any Canadian groups or company in oncology, and I think the hype is higher than the true PubMed impact factor that for me is the most important. Nevertheless, I think that we need to continue, we need to pursue, and over the next few and then years and not months, we will be able to reach an agreement and definitely use this platform to help our patients, to give access to big data, and I think we have to stop doing what I do, is to have one omic and to do a multi-omic trans-kingdom analysis is the future of at least oncology and I think of medicine, but it will take a lot of times.
That's going to be the word of wisdom of the closure, I think, because we've reached our time slot. So I'll turn it back to Stephanie.
Thank you, Anne-Marie. I think the discussion is coming to an end, and thank you to all panelists for taking the time today out of your busy schedule to participate in the roundtable discussion. Special thanks to Anne-Marie for moderating this very interesting discussion about sharing best practices internationally. If you would like to continue the discussion, please feel free to reach out to other panelists, depending on your interest, of course. The contact info details were provided in the package that was sent prior to the meeting. And on behalf of the organizing committee, again, thank you all for your active participation to the panel, and have a great rest of your day.
Thank you.
Thanks.
Thank you.
Bye.
Bye.
Bye. Bye. Bye.